Thanks for coming to our talk by Dr. Elizabeth Madden, who's based at Macquarie University in Sydney. May I call you Elizabeth? Yeah. Elizabeth is a postdoctoral fellow with World Wildlife Fund, WWF, and the Australian Research Council. She's going to be speaking about monitoring coral reef health from space. On a personal note, we met last year when we were starting to get ready to launch Google Earth Outreach Down Under in Sydney, and we were looking for cool nonprofits and scientists to showcase their work, Australian researchers. And we heard about her, and then meanwhile, through her network, she was hearing about us, that we were coming down there. And so we got together, and Elizabeth gave a wonderful presentation about this work at our launch, and we thought, wouldn't it be great if we could bring her back here so you guys could all hear about, well, monitoring coral reef health from space, but I think what's very interesting about it is not just looking at landscape features, but actually being able to observe marine animal behavior and infer characteristics of predator-prey relationships from Google Earth satellite imagery. So thank you, Elizabeth. Take it away.
Well, yeah, as Rebecca said, I think it's really turned out well to be able to come here. Thanks for having me. I'm really excited to be here, especially because so much of this work is based on using Google Earth. In fact, as you'll see, almost everything. So as she said, I'm going to be talking about monitoring coral reef health from space and the possibility of doing so with Google Earth. So as you all probably know or probably have heard, coral reefs around the world are in trouble. And that's due to all kinds of things, more global scale threats, such as sea surface temperature increase and ocean acidification, right down to more local scale threats, such as pollution, coastal development, coral mining, and destructive fishing. And one of the most insidious threats that is operating both at a local scale, because it happens at a local scale, but also at a global scale, because it's occurring almost everywhere on reefs worldwide, is overfishing. And that essentially is just taking out more fish and organisms than can be replaced. And what happens is normally when we take out fish or other organisms, but mainly fish for human consumption, we take the predators first. And by predator, I mean fish that eat other animals. So as you can see here, there's a tuna that's actually probably a dead fish. But it's, going and basically being a predator. And then you can see that, a lot of that's a big tuna market, presumably in Japan, I'm not sure. But just a shot from a sort of standard fish market where most of what you see is predators. So when we overfish coral reefs, we often first end up depleting the predators, which can really change the ecosystem dynamics. One of the other features of reefs worldwide is that lots of them are very isolated. So these are 3 reefs that I worked in during my PhD research. The Line Islands, which are part of the Republic of Kiribati and part of the United States. And you can see they're right out there in the middle of the Pacific Ocean, about halfway between California and Australia, so in the middle of nowhere, which has been great in many ways because historically people didn't go that far to fish. But nowadays what's happening is in this particular island chain, people are being moved from, or choosing to move from the main Republic of Kiribati Islands, and they're being brought out to the bottom 2 islands, Tabuaran and Christmas, to alleviate the overpopulation in the main islands of humans. But when they come to these islands, then they need food, and it depletes the predator and other populations on the coral reefs. The top island, Palmyra, is, interestingly, it's a reserve owned by the United States, and it has never really experienced a huge amount of, or a large amount of historical fishing. So you've got kind of a gradient of fishing pressure. But the point is that these islands are so remote that it's really hard to monitor them for reef health and understand what's going on. If you do want to do that with traditional methods of going out and swimming around, which I and many other people have done, it's very costly and very time and labor intensive. So it's hard to do. So this is kind of a problem that we have, and I'm hoping that what I'm working on now, what my colleagues and I are working on, can help to solve this problem. So I'm just going to give you a little bit of background on the sort of natural history and the ecology of what I'm talking about. Then we're going to look at the results that we have to date, and then I'm going to talk some about the next steps for this work. So some work that my colleagues and I did a few years back shows that fishing that is known to lead to loss of predators in coral reefs can also change the way that prey behave, the would-be prey of those predators. When the predators are gone, they become bolder. So what you see here is just this little blob represents a coral shelter, which is a safe place for a small fish. And this little small fish that you see is going away from that to eat seaweed. So that means it's a herbivore, it eats plants. And what we found is that when we took out when people take out the predators for fish and for food and other things, in a very simplified sense, it creates fearless small fish because they don't have to be as worried about being eaten, so they can go where they like on the coral reefs. What we also found is that you can, by changing the behavior of small prey fish, we as humans are inadvertently also changing where seaweed grows, which is why we have these dotted lines. Those represent indirect effects of humans on these things. So we're creating different patterns of seaweed distribution on the reef, which is important in its own right, but also because we believe that this may also lead to changes in where coral can grow. And obviously corals are the sort of backbone of coral reefs. Without them, there won't be any reefs. Yes?
What's the timeline for that sort of behavior change? Is that one generation or some?
That's a really good question. We haven't done any kind of looking at the evolutionary side of things, but it's definitely happening on ecological time scales, so scales within the lifetime of, I shouldn't say a lifetime of one fish, but within a few generations. So it's not necessarily an evolved response, although it might be as well. But what's happened is within these islands that I mentioned, humans have been inhabiting this sort of more populated islands over the last, I guess it's a few decades, really. And so these changes have definitely happened over that amount of time because we compared the areas that had no people and those that had people. So it's over relatively short time scales. And actually, the interesting thing about behavior is that changes in behavior can happen on instantaneous time scales. So it could be any range, really. So I'm not actually going to talk about the coral stuff today because that's something that we'll be looking at in the future. But just to give you a bit of an idea, is that really, to summarize, if you look from the top down, humans are changing how many predators there are in the ocean, and this is inadvertently changing things that we probably wouldn't have expected to happen, like prey fish behavior and where seaweed grows. So what we did is we developed a little model a few years ago, a very simple model based on a random walk, where this again is a coral shelter here in the middle, and a small prey fish goes out from that to forage for seaweed. So at each time step in the model, it goes out and comes back every time a predator passes by. And what that then does is it creates a probability distribution of foraging intensity about the coral shelter, which basically says small fish are spending more time foraging in the area of shelter, which is not surprising, right? So when you put that down onto a two-dimensional reef landscape and you kind of randomly plop these shelters down, what you see is that there are these safe areas that surround the corals and more risky areas where the fish can't get back to the corals to seek shelter. So it's a bit more risky to be out there and eating. The model then predicts, again, not surprisingly, that seaweed should grow in these risky areas more so than it grows in the safe areas. So you just get less seaweed surrounding reefs than you do in adjacent areas. So then, if you put this onto a slightly more realistic two-dimensional landscape, you see that there are embedded areas of safe zones within the risky zones. And those rings are basically should be bare of seaweed, according to the model's predictions, because they've been grazed by these small fish so heavily. Yeah, not so much. I mean, it depends. Like, for example, a kelp forest would offer some large, structural seaweed like that would offer some degree of protection, I imagine, from very large predators. But mostly in these systems, we're talking about on coral reefs, it's small, not very tall seaweed. So if you're like a medium-sized fish, you're going to be bigger than that anyway. So, and it's really no barrier to predation. So what this is exactly what the model would predict. This is a screenshot from Google Earth. And so you see these are the reef areas, these dark patches in the middle of the circles. And then you get these safe zones surrounding them with the risky areas in between. And what we presumed when we first saw this is that these risky areas are covered in seaweed and that the safe areas are what we would call a grazing halo. So grazing halos have been known for a really long time. In the Caribbean, in the late 60s and early 70s, they were documented and shown to be occurring within seagrass beds. So seagrass is just usually not that tall, really. And being driven by parrotfishes, which you see here, the sort of orangey green fish, and surgeonfishes, a sort of purpley fish. But another study came out a few years later saying, well, actually they're due to urchins, what you see here. And I think probably they're both exactly right, and it can be due to lots of different things. Basically anything that eats seaweed and is afraid of being eaten itself.
Are the same predators for the fish also eating the urchins?
It's a good question. Probably not. It would probably be a different suite of predators. You'd need a very specialized predator to eat that kind of urchin with those spines, any urchin. And these two fish species are pretty large generally, so they would require a pretty large-bodied predator. Yeah. And so, just taking a moment, what was cool about this is when we realized, hang on, we can see these things from Google Earth. We realized there's actually all this other cool stuff going on, which I'm sure you guys know so much about. But the neat thing about Google Earth and the problem with reefs is that it allows examination of these landscape scale features in really remote and really inaccessible places, which pretty much are where a lot of coral reefs are. But just a couple other things I came across that I thought were really neat and kind of relevant. For example, people are doing a similar kind of thing in Jordan, looking at the looting of historical sites. And basically what they found is by looking at Google Earth, they could see all these pockmarks on the landscape. And they realized that there was a much bigger problem than anyone realized with looting. And all the pockmarks were basically grave robbing. And so the outlines of red here are indicating where those are. And that's kind of like what we're seeing, which is large-scale changes in landscapes that you can't really see when you're just at ground level. Another one that was kind of cool, I thought, was looking at a study looked at Guantanamo Bay and used the time slider to look at pictures when there was apparently no official word that it was being built out more. But in fact, by looking at the time slider between 2003 and 2004, you can see that was a big difference. So that was just one interesting thing, sort of a digression, but I thought it was kind of neat. But what's cool is, so when we realized we could see these things from space, I'll just zoom you in so you can see the same thing. So we're going into Australia's southern Great Barrier Reef. It's a bit grainy, but, and this is Heron Island right here that we're zooming into. And within the lagoon of Heron Island, you get a lot of these patch reefs, these brown areas here. And what you can see is there's sort of a pale contour around the patch reefs. And on a very isolated patch reef, it's even more easy to see. But what you see here is essentially the safe zone around that reef. And then this greenish area here represents the seaweed, or so we thought. But we weren't sure, even though all the pieces made sense. But we thought, well, we better go and check it out. So now I'm going to tell you a little bit about what we've done with this on the ground. So my colleagues and I went to Heron Island and actually walked out into the lagoon in waist-deep water for about 1.5 kilometers to get to the closest of these halos from the island, because this is the island here, and this is the lagoon, and it's a pretty big lagoon. So we went out there with a handheld GPS. We got the GPS coordinates off Google Earth, put them into the handheld GPS, and just used it to get us out there. And lo and behold, we landed in the right spot. So what we did then is, this is just, again, showing you kind of the scale we're talking about here. The little yellow box corresponds to the next picture. And this is what it looks like. When we got out to the reef, this is what we found. These little patch reefs are actually not individual corals, like I showed you on the model, but groups of corals. So basically clusters of safe havens. And so you can see there are fish in here, lots of little things living in there, including some seaweed, but not the yummy kind of seaweed that you see outside of the reefs. And so if you just start to read this like a book, basically what it shows, it's pictures that we took. And this is a 1 meter squared quadrat. We just put it on the ground as we moved from the reef's edge out into that safe zone, into the grazing halo, and towards the seaweed. So if you read it like a book, that's 0 meters. That's right at the reef's edge. It's almost bare of any sort of seaweed. 3 meters, 6 meters, and so on as you move away out to 30 meters. What you can see is that it's a lot more green out at 30 meters, and that's presumably because of the fear that little fish have of going that far from the shelter of the reef. So then just to look at it a slightly different way, we have the distance from the reef edge here on the x-axis. So starting with 0 there at the edge of the reef, moving out towards 30 meters away. And then on the right-hand y-axis, what it shows is canopy height. And that's just a measure basically of what you saw, how tall the seaweed is there. And as you can see, the bars indicate that as you move further away from the reef, the algae gets taller. Not surprising. So then what we did, though, is that shows us what's happening, but it doesn't show us how it's happening. And as I said, we presumed that this would be due to seaweed-eating animals. And so we took little bits of the seaweed from the risky areas and clipped them onto clothespins, which we then weighted, very high-tech stuff, weighted to the ground with dive weights, And we just left them out for a couple days and saw what happened. And when we came back, sure enough, what this shows is the percent of the algae that was consumed. So the black dots show how much was eaten at different distances from the reef. And as you go farther from the reef's edge, farther from the safety, less was eaten. So that indicates to us that indeed it is due to little grazers that are doing this. We still don't know who. We'll get to that in a minute. So just to go back to the expectation then, here's what I showed you before. We expect that in areas that have less fishing and more predators, you get smaller safe zones. And that in areas where there's more fishing, and so there are fewer predators because we've taken them out, you'll get larger safe zones. And this expectation is what's led to the idea that we might use this as a tool to monitor reef health. So the goals of the project now are to determine if and how fisheries and marine reserves can lead to changes in coral reef landscapes via those cascading behavioral effects of humans, predators, behavior, and then seaweed that I showed you before. And if this is the case, we want to be able to develop a low-cost, global-scale tool for remotely monitoring these effects, and specifically on shallow coral reefs. But I should mention quickly, before I go on, that no-take marine reserve and no-take reserve, they all mean the same thing, and that just means that no fishing is allowed. So those should be the areas where there are more predatory fish. So what we predict is that grazing halos will be more defined in no-take marine reserves versus fished areas, because of course there should be more predators in the reserves, and also in older versus younger reserves. And this is important because when reserves are newly implemented, it often takes a while for predator numbers to come back. And so this might give us a first cut indication of when that's happening. And so if this is all correct, then it could be used as a tool to both remotely monitor the effectiveness of marine reserves as they are, and it could also be used as a way to help us identify areas that might be in need of protection. So areas where, for example, like those islands I mentioned, where there are more and more people coming, probably there's going to be increasing fishing pressure. And so maybe we can use this method to kind of indicate where those areas are that might be in need of some sort of protection. So what we've been doing is using the Great Barrier Reef in Marine Park in Australia, which is outlined here in red, as a sort of test case. And so that area, as you can see, spans a long area. It's about 13 degrees of latitude. And within there are adjacent no-take marine reserves and fished areas throughout. So it makes a good test case of looking, can we look and see, is what we predict going to happen? And the other thing is that it's very well managed in that the green zones really are protected from fishing in most cases, whereas a lot of places in the world you might get a protected area, but it's not actually enforced, so actually there's a lot of fishing going on there. So this is just a little slice from a zoning map there that you'll see, and what you can see is it's what's called a mixed-use marine reserve or a marine park. So you have a mosaic of green zones, which are not fished, and blue zones, which are fished, all throughout that whole area of red that you saw in the previous slide. And what we've done is we've been running these cross-shelf transects, running out from the coast of Australia here, following the lines of latitude and surveying through satellite imagery, through Google Earth, all of the reefs that we run into. And I should point out that when I say reefs, there are patch reefs that the halos form around, but those are all embedded within these larger reefs. So these areas are all reefs. They can be a few kilometers long. They can be much larger than that. So you get an idea from the lines of latitude there, how big they are. And so when we're doing this, this is looking within one larger reef. These are little bitty individual patch reefs and the halos that are surrounding them. So what we're doing is we're measuring characteristics of halos like reef, larger reef, smaller patch reef, and then the halo size and area, the halo width and rugosity, which is basically just the wiggliness of the circumference. And if no halos, what's the reason? Because in a lot of cases what we're finding is that there are things like surface reflectance or the image quality just isn't high enough to be able to get at these things because of the scale over which they are. So if there are no halos, what's the reason? And again, the expectation here is that the no-take reserves should have smaller halos, and the fished reefs, where there aren't as many predators, should have larger halos. So this is a cool little video that Tyler and some people here put together. Thank you again. And it just gives you an idea of going in. So we're looking at Australia here as we're going to go in. Let's see, I'll play it here. As we zoom in, we'll see a larger reef. Oops, it doesn't seem to be going. Let's see if it goes. Oh, doesn't seem to want to. Anyway, it's okay, we can move on. But you've seen, you get the idea that there are larger reefs, and within that there are smaller reefs, smaller patch reefs, and that's what the halos form around. So these little pins represent all the reefs we've surveyed with this method so far, and it's about 1,200, which is pretty time-consuming, but it's generated some data so far. So what you'll see is, so we started with this many, but then when we get down to the reefs with visible substrate, or visible benthos, which is the reef floor, because of those issues I mentioned before, it really cuts it down a lot. So it's about 200 out of all those 1,200 that we can actually see down, and the image quality is high enough that we can look for these halos. Then, when you look again and you look for the reefs that have halos, only about 50 of those have halos. And that can be due to a variety of reasons, but it requires a very specific type of habitat, these patch reefs, and not all reefs have that kind of habitat. So you wouldn't expect it in all of them, but it's also useful to know where there are reefs with those that type of habitat that aren't showing halos. So the first little analysis we did was just pool them all together and see if there's a pattern, a difference between green zones and blue zones, the fished reefs and the no-take reserves. And so on the y-axis, what you have is halo radius, which is just the size of the halo. And we found that, contrary to what we were expecting, there really wasn't any pattern, or if there was, it was slightly in the wrong direction. So that puzzled us for a minute, but not very long, because when you're working over this large of an area, there are going to be latitudinal differences, environmental differences over that latitude that could affect the size of halos. So what we did then is we paired them up, and we said, okay, let's remove all the variability that comes with an environmental gradient like that, and just look at green zones, just compare green zones that are not fished and blue zones that are fished that are adjacent to each other or close by. And so when we did that, we ended up with 8 pairs, which was, as you can imagine, kind of disappointing because we've done all this work. So, oh, we've got to have more than that. But nonetheless, we went ahead and looked, and what we found is that the pattern was very striking in the direction we would have expected when we took out all of that other sort of variability. And so again, you've got the halo width on the y-axis, and we've got our fish reefs with larger halos than the no-take reserves. But we also realized that, so we saw the expected pattern, but by chance, what we also found was that for some reason, I mean, because it's a small sample size, variability within the reefs is going to have a big impact. We found that there were some larger patch reefs in the blue zones, the fish reefs. So that could be driving the pattern. And we also thought about, well, what about the effects on halo size of things like density of the small fish or invertebrates that could be making these halos? And therefore, they might have to compete and go farther from the reef to eat, to get their food, so on and so forth. And just generally in reef area might affect the size of these halos because they're around a patch reef. So we then took that out. We standardized the halo width. You can see on the y-axis, it's now halo width divided by patch area. So we tried to standardize by the patch area, the small reef area. And we found that, again, the pattern still held. And actually, this is a highly significant result because of the test we use. It's very good at picking up small differences, even though there isn't apparently a very large difference in the average size of these halos. So that was encouraging because the pattern remained, the expected pattern we saw, the pattern we expected was what we saw. So this gives us some degree of confidence that, that this is a real phenomenon. Yes? Yeah, it does. I know, actually, I'm going to look in, this is all brand new, basically, but so I'm going to look into this a little more to figure out what's going on, but definitely the result is it's highly significant. So I think I've got to actually just look at how the error was calculated for the graph, but when you actually do the analysis, it's a repeated measures test, and it comes out highly significant. So, and when you look at it, you just eyeball it, you can sort of see the same thing. Okay, so now I'm just going to talk briefly about the things that we're hoping to do next with this. So obviously with 8 pairs, we're looking to increase the sample size. That's the first obvious thing to do. So what we need to do this is we need more high-resolution imagery at 1 meter sort of resolution or less. But so thanks to Brian and other people here, there's a request in now to GeoEye to get some of this imagery. So Brian's helped us request 94 scenes, and these are some of them. that will hopefully allow us to overlap with some in situ fish data, some data that actually exists on how many predators are there on some of these reefs. There haven't been many that have been surveyed in detail, but there are some, and we're hoping to overlap with those. It will also allow comparisons over time. So as I mentioned, the Great Barrier Reef Marine Park was rezoned in 2004, and this will allow us to look at how the new reserves are going over time. Are the predators recovering, and can we tell that from this information? So thank you again for helping us with that, because really we wouldn't be able to progress past this point without some more high quality imagery. The next thing we're going to be doing, and we're already working on this, I've just gotten back from Heron Island a few weeks ago, the first island I showed you, where we are trying to understand the mechanisms of halo formation. So what's going on? Who's making these halos? Is it the fish and inverts that we think? What predators eat those species? And then importantly, do we eat those predators? Because if that's not the case, then there's a disconnect. So we need to figure out what's going on here. So what we've done is we've put out these little GoPro cameras in the reefs, the patch reefs here, and just out in the halos where it's pretty much bare sand, and just left them running. Because when you're out there yourself, you really don't see much. It looks like a barren wasteland almost. But it's pretty cool because we thought, oh, it could just be a lot of empty video. But what we're finding is that there are actually some things showing up, and I'll touch on that in just a minute. But essentially what we've done is if you've got your patch reef and your halo and your risky areas out here, we've just set up an array of cameras kind of moving out from the reef itself out into the seaweed areas. And each one of those numbered boxes is a camera. So we left those out for about 4 hours every day over a period of about 3 weeks and got about 20-something reefs done. So hopefully we'll get some good information out of this. Like I said, it's brand new as well, so we haven't even begun to look at the data. But when we were downloading it, we did get some just great little shots of things. These are spadefish or batfish, and they're known. Actually, we've never seen them out there when we've just been swimming around, setting up the cameras and doing whatever. Never come across them, but apparently they're there. And they're known to be very voracious herbivores that can really decimate a whole bed of seaweed in a short amount of time. So if they're there, that suggests that they may be part of what's going on. Now, we'll see if this works. Oh, yeah, it does. So these are some surgeonfish, which are one of the two groups that I showed you earlier that had been implicated as doing the halos in creating the halos in the Caribbean. And so we never had seen these out there. We'd seen them in the little reefs, but we hadn't seen them out in the halo areas. And apparently they're out there eating. So also there are predators out there, which hopefully this will come. Yeah. So here are some sweet lips. These are mainly predators of smaller fish and invertebrates, but they're called sweet lips. These guys would be about this big. And just seeing that they're out there in these halos means that there is a risk for small fish. So at least we've got a couple pieces of the puzzle now that we know that there are prey out there eating and there are predators. And interestingly, here's a lemon shark that we saw out there. And these are some more sweet lips. And we were swimming around out there one day and we came across this little school here. There were actually quite a few of them. And this one here, I don't know if you can see what's going on, but he's basically been sliced open by something. I don't know what. It doesn't actually look to me like the kind of bite that would come from a shark attack. I don't know what it could be. But there's obviously a risk for even larger fish out there. So there's some kind of risk because this guy got attacked by something. He was still alive, though, and pretty happy, seemed to me. So we know that there's predation risk out there as well. And this is kind of just extra because it was so graceful and beautiful. Just a stingray that kind of made a cameo right across one of our cameras. And there are lots of stingrays and other kinds of rays out there, which may even be part of the story. We don't know. It could be something a little bit to do with bioturbation and stirring up the sand, making it harder for the algae to grow. We'll soon find out, I hope. Okay, so then in terms of changes over time, like I said, one of the next things we're hoping to do is look at, go through the marine park again in the Great Barrier Reef and look at the reefs that became no-take reserves that used to be fished and now are not. And so look at halo size or presence before and after the rezoning and also over time after the reserves were established. And so the question there is, does grazing halo presence or size change when fishing starts or stops or as predators recover? And then this is where I'm particularly interested. This is what kind of motivates the whole project, is developing this conservation tool that I hope will come of this. The way we're going to do this is we're going to start by focusing on 3 of the World Wildlife Fund's priority areas, and they've identified some areas around the world that are in urgent need of conservation attention and also have a high conservation value. So we're hoping to do some sort of more detailed look at these places and work with the local people there, utilizing local knowledge and expertise and kind of building that into the tool, and to develop then and test this tool in a few targeted places. And once we've done that, oh, and the areas where we're going to be working for this are the Coral Triangle, coastal East Africa, and the Mesoamerican Barrier Reef in Belize. And once we've done this, we're going to be feeding that in, as I said here. So the idea is to kind of package this thing as a tool that really anyone could use anywhere in any coral reef system in the world, scientists, managers, whatever, and package it with kind of Google Earth. It would be more or less the backbone of it. And then we'd put it together with the sort of the know-how, the statistical protocols and the data extraction protocols, and then always have a way to build in local knowledge, because every system is different. And although we might find that in Heron Island's case, it's species X, Y, or Z that's making these things, we know it's a global pattern because we've seen it everywhere, but the same species might not exist in every system. So we need that local information to build into it and figure out who are the species that are really playing a part in this in different systems. And then I'd like to then take it global and kind of look at the entire tropics. So from about 27.5 degrees south to 28.5 degrees north, looking at both the spatial and the temporal effects we've seen, and then test it in a whole range of diverse systems. Because when you use Google Earth and just have a look around, you see these grazing halos everywhere, from the Red Sea to the Caribbean to Great Barrier Reef, all over the place. And so I think that there's definitely some potential utility to use this as a global scale tool. And the idea is that it will be a remote tool for resource management and conservation. So, for example, this is an island off the coast of Jakarta in Indonesia. I don't know if it's clear to you what's going on here, but basically this is an island that's just covered, every square inch is covered in roofs. So you can imagine the number of people living on this small island and the pressure on the coral reefs in terms of fishing that would be happening, because everyone needs to eat, right? So because this is an island, one of many, it's actually in a chain called the Thousand Island Chain. And it's unlikely that any country, Indonesia or any other country with limited resources, which is every country for monitoring these things, would have the resources to monitor this and every other reef like it. So this is just a great example of where we need a tool like this, because there is probably a lot of fishing pressure, and the reef may be changing rapidly, but unless we monitor it, we won't know that. So I guess the key novelty with this method is that it can be, we can look at larger areas and more of them in less time and at lower cost than we can do otherwise. It won't ever replace in situ. We'll still have to swim around and do that sometimes, but I think it would allow us to do a much broader approach to monitoring reef health. So with that, I'd just like to say thanks for having me, and thanks for the support, because also we wouldn't be able to do all this without the sort of polygon measurement tool in Pro. And so having it really does make it a lot quicker. Otherwise, we have to do it on a digitizing tablet and blah, blah. So it's been great having that, and just having the support of you all helping us get the better imagery that we need is great. So thanks very much.
So what does reef health mean? Why is it important that tiny little fish stays in the reef itself?
Okay, so essentially when I talk about reef health, predator populations are one aspect of reef health, because as I said, when we fish reefs and we take out the predators, that changes the ecosystem dynamic in ways that we now know. There are some surprising ways, but I'm sure there are many more. So as we change the food web structure of the reef, we're changing, I mean, you could call it health. Health is a very simplified way of saying it, but predator populations are indicative of one aspect of reef health, because if we lose all the predators, then it changes everything below that, and we don't really know what's going to happen. And so, as I said, if you get right down to the very bottom, it could be that it's even changing where coral grows. And if we change where and how coral grows, then we're really altering the fundamental structure of what's there. And without corals, there would be no reefs. No, so they control, these tiny fish control, and probably invertebrates too, control where the seaweed grows. And it is a little complicated, so I apologize for that. But where the seaweed grows, if there's a thick mat of seaweed, it's very hard for corals to grow there often, if it's certain types of seaweed. And so if we change the abundance or distribution of that seaweed, then we could, I mean, this is all hypothesized. As I said, we haven't tested out the link between seaweed and coral. I mean, it's a known link. but we haven't tested it out in this particular case if it can be an indirect effect of humans. But the theory is all there, and it makes logical sense. So even without that aspect of it, the point is that by changing coral reef ecosystems in ways that we don't really understand all the ramifications for, we're going to change. It could be potentially quite bad.
I wonder how you distinguish between the larger grazing halos being due to fear versus maybe just larger populations of the fish when there was no predator.
Yeah, so that's a really good question. And that's one of the main questions with the mechanisms that we're trying to understand now. We're trying to understand, by doing these video surveys, we'll be able to get an idea of, are there more fish in the halos than there are outside, or we'll just have a better idea of what's going on out there in terms of both numbers and fear, although it won't give us much information about behavior. So we're trying to basically get a snapshot, or a video shot, I suppose, of what's going on out there. And from that, we can start to piece together and understand, could it be due just to numbers, or could it be due mostly to fear? And yeah, based on the theory and what we've seen, I feel fairly confident that it is a fear thing because, well, there are a variety of reasons which we can talk about later if you're interested. Yes?
What kind of job do the reserves do of actually protecting the predator fish? Do they travel through a large enough territory that they're likely to swim out of the reserve and get caught?
Yeah.
If you have these reserves adjacent to fished areas?
That's a really good question. And that's a term for that. It's called spillover. So, and it's actually great for people who want to fish because fish don't know the boundaries of reefs, of course, so they just swim right in and out. But it's a probability thing. So if you are protecting, you could even think of it as time. If you're protecting more of their time of an individual fish, there's less likelihood it's going to get caught. So over a population, on an individual level, it doesn't, individual fish level, it doesn't have a whole, it doesn't mean that much, but on the population level, it does. Yeah.
Have you looked at doing any studies for the foraging fish behavior with fish tracking? Like you've done this with cod, where you put in a pen and then you put trackers on them and you watch them and look at their behavior.
Yeah, and that's one, I mean, a lot of people have done that, and actually we're hoping to work with some people who have some predator tracking data for the Heron Island Lagoon, so we can see, actually it's for tiger sharks, and see where they go. But the problem with doing that for small fish, and some people do, but in a system where there are a lot of predators, then it's actually happened to a colleague of mine in Palmyra. They started they started capturing fish and doing a few tests, taking some blood to see how their cortisol levels were and such. And just by manipulating the fish, so if you wanted to put a tracking device in, say, you'd have to manipulate the fish. And then when you put them back in the water, they're suddenly much easier to catch for a predator. So some colleagues, actually, what they ended up doing is they were trying to track bonefish in Palmyra's lagoon. And so they would, I don't remember how they did it, but they captured them, did the stuff, put them back in. And they were finding these really unexpected patterns where they would see like these big movements that they weren't expecting for the bonefish, and then they would just stop, and then they would just sit there. And so finally what they figured out is as soon as they threw the bonefish back in the water, they were being eaten by sharks. So they were actually tracking sharks all around the lagoon. And then the sharks were pooping out the pinger things. So, and that's why they stopped. So we could do that. That's probably something that we'd like to not have to do if we can, because it has a lot of trouble associated with it. But it's a great way to figure out the movement, yeah.
Way to track predators without some stress on that.
I think they actually changed the project to focus on the predators once they realized what was happening. Tyler?
So in terms of your non-invasive monitoring with the GoPros, I don't know if that's the normal. You're probably not a typical market for GoPro, but they're more like 20 minutes away. They might actually.
Oh yeah, that's a really good point. Yeah. Like I said, we've just gotten this video data all back, and we finally just got it into one place, and haven't looked at it much, but I just from what we've seen, it definitely has some cool stuff. So maybe I should let them know what we're doing. Yeah, well, that was a limiting factor, is how many cameras can we get together? We got 20 at one time. That's a great idea. All we need then is the manpower to put them out, because it was such an effort getting them. Doesn't sound like it would be, but getting out there, putting them out, starting them, doing all the reefs, and then going back and doing them all and picking them up that day. But if we had a team of people, we could cover some ground.
Is that high resolution imagery that you're going to get? Are you going to try to do sort of object recognition on there?
You know, we've looked into that, and I would love to hear what you all think about that, because that is definitely none of the sort of GIS type stuff is where I'm coming from. I'm coming from an ecology point of view, so I'm always interested to hear your thoughts, but that's another thing. I have talked to quite a few people and tried to find out if there's a way to do that, but because, as you can see, it's a very fuzzy boundary, these things. It's not a very distinct, clear boundary between the safe zone and the risky zone, the halo boundary. So a lot of times it feels like more of an art than a science. And I showed you some clear examples, but there are also a lot of, in a lot of cases, what we're finding is the halos are really faint, but there are certain criteria you can use, which you could probably teach a machine to do, to understand. But I think the problem would be getting, I don't know, it might be possible. And I would love it if it were, because it would save so much time.
I would recommend talking to Imagery. I forget who it is. But
Okay.
somebody there has put out open source software for ROV video.
Okay, all right, well, I'll have a look into that. So Mbari, you said. Okay, thanks.
How much the seaweeds itself is really changing kind of over time? Because when you have a satellite imagery, you get a very clear snapshot, and you're like, here's where the halo is.
Yeah.
Are there spots that you're able to monitor that are close by you, to know how much that fluctuates throughout the year? So if there's like a nutrient rush or there's a sewage thing and suddenly everything blooms.
Well, that's a really good question, and one I wish I had thought a little bit more about before our last trip, because we were using, we've got the image, which I think current image on Heron Island is from 2006 in Google Earth. And it's very clear with the halos and whatnot. So we got our team together, we went out there, we had this plan for 6 months. But when we got out there, we found that there was actually not much seaweed there. And it turns out that I think it's a seasonal thing, which is not surprising. It's just that it had been so stable in the previous times we'd been, it had been there, so we thought it'd be there all year round. Well, it wasn't. But the pattern of the halos was still there. It was just much more faint. And again, this comes into why I think it would be hard potentially for a machine to do it, because it was very faint. But I think that luckily, not luckily, but the mechanisms that are creating these things are stable over time, I believe, because the ring does still exist. It's just a lot more faint. So yeah, it's definitely there's seasonality, and part of what we'll need to build into the model, especially with particular local areas, is understanding if there are pulses of nutrients. Like if there's, for example, on the Queensland coast of the Great Barrier Reef, is sort of a monsoonal season, and when that happens, all these nutrients are washed off from the watersheds and the adjacent land areas with sugarcane and things growing. So you get huge pulses of nutrients. That could definitely affect it.
You mentioned creating, an operational conservation tool. So if you want to monitor, how frequently do you need updates to have it actually be effective?
Well, that's a good question. And I don't know, really. I don't know. Like I said, I wasn't really expecting these things to change over the time scale of, months to months, but that does happen. So I don't know. I think if I had to guess, I would say yearly to 2 yearly kind of thing, maybe 2 yearly, because changes, especially reductions in predator populations. I mean, a boat could come in and just wipe out like a shark. That happens with shark finning boats. They come in and they go and they take tons of sharks off a reef, and that's it. One day later, that's changed. But it probably takes a bit of time for these behavioral changes then to manifest, and then to manifest down to the ring. So I imagine it would be yearly to 2 yearly, possibly, or I don't know. I guess we'll have to wait and see on that.
Do you have a sense for the new study areas you have, whether you've got the imagery there you need?
No, we haven't. That's sort of so far down the track that we haven't really gotten, I haven't really had a good look to see yet. That'll be, obviously, they're big areas, so hopefully my experience thus far has been that there are, within any given area of reef, you can find some patches of high quality, high resolution imagery. So whether that's enough or we would need more, I don't really know yet.
To get your own imagery, have you considered doing something like the quadcopters or balloons or
Like
kites?
drones and stuff like that? Is that what you're thinking of? Or?
Just to take up a typical camera, not the full-on drone sort of concept, but the cheap end of like a balloon or a quick get up there with a picture so that you could then geo-reference local pictures that you take while you're surveying.
Yeah, the thought has crossed my mind, but not in any cohesive way. So I'd love to talk to you more about that if you have some ideas of how that could be done, especially for specific areas. Yeah. You have some?
So your random walk model predicted like a smooth gradient of that in the satellite imagery we didn't see that in the halos and some of the halos. So what's the difference?
Well, the model is really very simple, and so it's going to predict, a boundary of a safe zone, whereas in reality, it's probably, these halos are probably driven by different species which have different thresholds of risk tolerance and things like that. So it is going to be a much fuzzier boundary in nature. The model was just basically to give us an idea of, is the mechanism that we're thinking that's responsible for these things, and it actually wasn't designed for halos. It was designed for the reefs at Palmyra, which don't have these patch reef characteristics. It's actually more continuous reef, and it was a bit perplexing. It was really hard to get our head around what would be the outcome of the seaweed if you've got reefs that are sometimes overlapping in zones, sometimes not. And basically it predicted that it would be more patchy, more heterogeneous than you would expect if the risk was kind of even everywhere. So yeah, it's not, the model does not necessarily map onto nature exactly.
Gradual change.
Yeah, in terms of where it goes from the sort of halo safe zone to a darker green, yeah, it's not a distinct boundary, it's a transition kind of zone.
Basically,
Yeah.
you cherry-picked the examples for
I
this
tried
talk
to show
that
you
were
very
most
clear examples. What you do see, though, is, you know, I think that probably in a lot of cases what would happen is the halos might get so big and that there's so little fear that they just merge and then there's no pattern whatsoever. But that's valuable data, too, because that's something we could compare then to, say, a reef that has more predators in the same area and see if we do see halos in one but we don't in the other. All right. Oh, yep. That's what we did this time, and some of them actually were more like 3 hours. It was limited by battery life, basically. And actually, no, I take that back. So we use some Hero, I don't know if you know the cameras, but some of the Hero 2s, and those were limited by battery life. But I brought a crop of Hero 3s, and they turned out to be limited by the data storage, because I guess you can't set the image quality to be lower than I guess the basic setting is, and that really sucked up the memory. So for the next time around, we'll have to get a bigger card, basically, get more information.
The full video, or could you do snap photos every 30 seconds or something else?
Yeah, we can definitely do that. And what we'll do is we'll just standardize by the amount of time that the camera lasted for any individual camera. And we may subsample, although I think I'm just going to hire someone to go through and have about 4 screens up, or maybe more, because it's mostly blank, like I said. And when something comes along, you know, quantify it, do the identity and the size and the number of the species. But I think it'll just be better just to go through, because the events of when you see things are kind of rare. It's a lot of just looking at water. So we don't really want to miss any if we can. We don't want to compact our sample size any more than we have to. Okay, well, oh, yeah.
Do you have any good data on how much anything's bounced back since the MPAs went into effect there, whether it be from your group or other ones?
Yeah, not from anything I've done in particular in the Great Barrier Reef, but there's one study, I think it's a group of people that have all kind of put their data together, and they found that within, I think it was 3 years of the reserves being established and starting to be enforced, because I think there was about a year grace period, they were seeing increases in the, I want to say, size structure of the predators, and possibly also abundance, because things don't reproduce they wouldn't reproduce and get to be big fish at that time, but because of the effect that you mentioned of crossing boundaries, it's basically just like an area where there's less likelihood of being taken out. So anything that crosses in there, you would be counted in the survey. So yeah, on a few years scale, but there have also been other studies that have been looking for years and still not seeing much of an effect. And it has it's probably a very context-dependent thing. So some areas, it might be slower growing predators, or what, I don't know. Sometimes the reserves are quite small, and so it's going to take a long time before biomass builds up in there. And on the bigger reserves, yeah, they're seeing effects pretty quickly. Well, thank you all very much for listening and your good questions.
beautiful slides.
Thanks. Well, thanks to you all.